Okay, this is my Chapter 11 PowerPoint on microbial control. So, about microbial control, it's been around for a long time, and they used to use salting, smoking, drying, or pickling, and sunlight exposure to preserve food. But now they are able to control microbes through more modern methods like physical methods. That includes heat and radiation, and then there's also chemical disinfectants. They limit contamination and infection. So there are different outcomes of microbial control. So sterilization, this is the complete destruction of all microorganisms. So this includes bacteria, endospores, viruses, completely gone. And this is used for like surgical equipment that need to be completely clean. And then there is disinfection. So disinfection, it targets vegetative pathogens, and vegetative means that it's just like actively growing and reproducing. So it targets vegetative pathogens, and it does not kill spores, but this type of microbial control is good for non-living surfaces. And then there is antisepsis. So antisepsis is also called degermination, and this is what's used on living tissues like skin or mucous membranes. So it's used on skin because we cannot sterilize skin. It's way too harsh. So antisepsis is the route for that. And then there is decontamination. And decontamination is also called sanitation. This just reduces the number of microorganisms that's on a surface to like a safe level. So think of like cleaning your dishes or kitchen utensils. So each method is different on its thoroughness because there's many different applications for each of them. So I'm going to go a little bit more in depth. So sterilization is an absolute term, meaning that it either is completely sterile or not sterile at all. So there's no ever just kind of sterile. It's either sterile or it's not. And disinfection is, you can either use chemical or physical means. So like think of bleach or boiling water to eliminate vegetative pathogens. And antisepsis, this is using mild chemicals, so like alcohol or iodine on like living tissue to help prevent with infection. And then decontamination or sanitization, this uses like soaps and detergents. So this is just physically removes microbes or dirt, debris. And sepsis means microbial growth in tissue, and asepsis is the practice of preventing infection and contamination. Oh, and asepsis, I like to think of like a doctor washing their hands, so they're just preventing the contamination. So then we have microbial resistance levels. So microorganisms, they are more resistant or less resistant to certain applications. So bacterial endospores, these are the most resistant, toughest. They require sterilization in order to destroy them. And then there's prions. These are infectious proteins, and they are even more resistant to decontamination, and they need extreme methods. Then there is the moderately resistant. So these include fungal spores, protozoan cysts, naked virus. So vegetative bacterial cells are also a part of this, and they're envelope viruses, which makes them more easy to be deactivated by like disinfectants or heat. So microbial death, it isn't always apparent because microbes, they don't show signs of death like an animal would. So death for microbes is the complete ability to reproduce. So this uses lethal agents like heat or radiation, and it causes damage to membranes or proteins or genetic material. So death, it occurs logarithmically. So this means the more time or concentration, the more amount of microbes cells will die. And sterilization, the goal of this is to reach a point where no cells remain, no viable cells are there. So factors that can affect microbial death rate. So there are many different factors. First off is a higher population. It'll require longer exposure, and there's also mixed populations like containing endospores or cysts. These are harder to eliminate because they're mixed. So then temperature and pH. This can enhance or reduce the effectiveness of the control. And then concentration. Concentration matters because stronger solutions, they're usually more effective, but they're also more corrosive. And then presence of organic matter. So organic material like blood or saliva, these can block agents and reduce their impact. And then lastly is biofilms. So biofilms, these are microbial communities like we learned before on surfaces. So because of the way they're made, they're especially resistant because they try to protect the biofilm. These are especially resistant. So there's physical and chemical methods of control. Physical methods include moist heat or autoclaving and dry heat. So these are effective at sterilizing like surgical equipment, any type of like instrument material. And then there's also radiation, so like gamma rays or UV rays, and these can destroy DNA. And then there's also chemical methods. So chemical methods include like disinfectants like bleach, alcohol, phenolics, and these are used on surfaces typically. But then there's antiseptics like hydrogen peroxide that's used on living tissue. And then there is also chemical sterilants. These are like ethylene oxide gas, and they destroy endospores, and they're also very toxic so they're carefully used. So antimicrobial agents, they like to target different components in the cell. So some disrupt the cell wall and will weaken it and cause lysis. Like surfactant damage membranes, they make them leaky and destroy selective permeability. So then agents that affect nucleic acids are like radiation or formaldehyde. These prevent replication and transcription. And then others will denature the proteins or enzymes, which will halt metabolism and keep it from happening. So these methods, they're selective, like heat and radiation. They kill a different variety of microbes, but there's also more specific agents that'll just target certain structures in the cell. So I'm going to talk about the different physical microbial control methods. So physical control methods, these rely on a changing environment. So like conditions that can destroy or inhibit microbes. So there's heat, cold, radiation, and filtration. Those are the most common methods of physical control. So they target vegetative cells, spores, viruses, and fungi that are usually found in like water, food, air, or like medical equipment. And heat is the most widely used because it's more reliable and it'll denature the proteins and it'll damage cell membranes. But there's also cold methods like desiccation, radiation. They're used because they're very effective, and the goal of this is to either sterilize it and eliminate all life, or just reduce the number of microbes to a safe level. So heat as a control agent. Heat kills microbes, it denatures proteins, disrupts membranes. We've seen that before. So high temperatures, they destroy enzymes and structural molecules, and it leads to cell death. So there's moist heat, which is like using steam or boiling water, and this is efficient because water will conduct heat better than air. But then there's also dry heat. This is like ovens or flames, and they dehydrate and oxidize the components within the microbe. So the temperature and time of exposure will determine how it's sterilized and or where disinfection occurs. So moist heat versus dry heat. Moist heat works at like 60 degrees Celsius up to 130-ish, 40-ish degrees Celsius. So it causes proteins to coagulate and denature, and it'll stop the metabolic processes super quick, and it's more effective because it's more effective at lower times and lower temperatures and shorter times. So then there's dry heat. So dry heat is way hotter, so like 160 degrees Celsius up to like over 1,000 degrees Celsius. So this kills through dehydration and oxidization. And you would think that dry heat would be more efficient because it can get way hotter, but actually it's less efficient. And flaming loops or incinerating waste are examples, so both methods can sterilize if it's properly used. So bacterial endospores, these are the hardest to kill because they survive boiling and they require higher temperatures. And then vegetative cells, these are like fungi, bacterial cells, protozoa. They die easily. And viruses, they show moderate resistance. So they can survive like 55 to 60 degrees Celsius for a long time. So it's basically effective sterilization. I'm sorry, that's my cat. Effective sterilization is like a good balance between time and temperature. And then there's 2 ways to measure it. Thermal death time is the shortest time to kill all microbes at a set temperature. And then the thermal death point, this is the lowest temperature that kills all the microbes within 10 minutes. So now I'm going to talk about the moist heat method. So this includes boiling water, pasteurization, and steam under pressure or autoclaving. So boiling, this kills most pathogens, but it does not kill endospores. So it makes it a better fit for like disinfection at home. And then pasteurization, this is like a mild heat, and it is good to reduce microbial load in like liquids, so like milk, juice, beer. We've heard of this in the past, pasteurization. And then autoclaving. So this sterilizes at high heat, like 121 degrees Celsius. So pressurized steam allows temperatures above boiling, and this is like the best way in labs and hospitals because it destroys endospores and all microbial life. So then we have dry heat methods. Dry heat methods include incineration or a hot air oven. So incineration, it completely destroys all microbial material through combustion. So this is good for like lab instruments or hazardous waste. And then there is hot air ovens. So this is like convection heat. It goes for longer at high temperatures, and it can't sterilize items that are like plastic, so it's only limited to a certain amount. So it also, we said this before, it seems effective. Dry heat requires a lot more time and energy though. So talking about cold and desiccation ways. So cold temperatures like refrigeration or freezing, they just slow down microbial growth, but they do not kill them completely. So pathogens, they can survive for months in a fridge, but desiccation, on the other hand, it removes moisture and inhibits the growth. So it doesn't really kill all the microbes, and they usually remain viable. And then lyophilization. This is combining freezing and drying to preserve like cultures and samples for a long time, like even years. So there's different types of radiation control too. There's ionizing and non-ionizing. So ionizing radiation, this includes like gamma rays, X-rays, and it will penetrate and sterilize materials that can't handle heat. So like medical equipment and packaged foods. So it causes DNA mutations and it destroys enzymes that are used for repair. But then there is non-ionizing radiation, and this uses way less energy. So UV light, it forms dimers in DNA, and it prevents replication. So but the downside to it is that it doesn't penetrate through glass or solids well, and it can also damage human tissue. So then there's filtration. Filtration physically separates microbes through, like, they use either fluids or air. So liquids will pass through membranes with tiny pores that help trap this bacteria and some viruses. And this is good for sterilizing heat-sensitive solutions like vaccines, IV fluids, or other types of media like that. And the HEPA air filters. These remove airborne contaminants in like hospitals and labs. So filtration, it doesn't remove toxins or dissolve molecules, but it is reliable for producing a sterile product. So we're going to talk about osmotic pressure, and this is osmotic pressure control. It uses salt or sugar and to draw water out of the cells and causes them to shrink and die and stop growing. So this is a long time ago, a technique from a long time ago, like used in like jams or like cured meats.